class. This is your lab exercise on the ABO blood typing system. So please check out this resource down at the bottom, kind of a lots of good information there just from the American Red Cross. That's actually where I got a lot of this content from. Okay, so we're going to do a PhysioX activity on blood typing. But before we do that, let's talk a little bit about the basis of the ABO blood grouping system. So first of all, we're talking about the carbohydrate antigens that are present on the cell surface of red blood cells. And this is important in transfusion reactions or any type of transplant because we will react to the wrong type of blood. But what does that mean? So let's go through it. So first of all, we've got to talk about the genetics of this a little bit. We've got two different patterns of inheritance that are going on with the ABO blood grouping. So just to remind you, dominant pattern of inheritance means that even if there is one recessive allele, the phenotype will be that of the dominant allele. Where codominant is where two different alleles, both of them are expressed at the same time. That's what it means, together. So A and B are encoded by the genes for either A or B. So we have three different alleles in the human population that are part of the ABO system. So it is I to the A, I to the B, and then little I, which we'll get to what this means. So the A and the B are dominant to I. So they will beat out I every time. I is recessive. But A and B to each other are codominant, which means they can be expressed at the same time. So let's begin by just talking about the four phenotypes that we have in the human population. So we've got phenotype A. So this person has blood type A. There's blood type B, blood type AB, and then blood type O. Now let's work backwards. Let's look at the possible gene combinations that can result in the phenotype of blood type A. And we actually have two different possibilities. A person can have both dominant A alleles, or they could have only one dominant A allele and the recessive little I for incomplete. So we've got two different genotypes that can specify the 1A phenotype. The same is true for B, but now we're talking about the B carbohydrate gene. So we've got big B with the incomplete, with the recessive form of the allele, or both dominant versions of the B allele. And then here's where we see the codominance is with the phenotype AB. So a person that has phenotype AB, type AB blood, they have the allele for both A and B. So they express both of these antigens on their cell surfaces of the red blood cell. And then the phenotype O, or the person that has blood type O, they have none of it. They have two of these recessive forms. So that's the genetics that result in the different, the phenotype, which is what's relevant really to physiology here. So let's move forward. We'll talk about the Rh factor at the end of after we get through this part of it. So right now, all we're talking about is a person being A or B something. Okay, so now let's look at this table here that summarizes what's going on in our different blood typing basis. So there's actually a lot of information here, and it may not be as inherently simple as you think when you look at it. So person with blood type A has the A carbohydrate on their cell surface. So in this figure, it is this, looks like a lollipop, a little purple circle lollipop stick stuck on it, a spike. B is the light green diamond, and then A and B would have both. So we've got the A allele, or actually it's not an allele anymore. We're talking about the protein. So we've got the A, we're talking about the carbohydrate antigen. We've got the A and the B, both of them are expressed. And then on blood type O, nothing's expressed there. They don't have either of those cell surface carbohydrates on their cell surface. So early in development, the immune system, as we are learning in immunology, the immune system develops really early. It develops during embryo, or it begins during embryogenesis, and then different parts of it continue at different times. So what I didn't talk too much about in our immunology lecture is a concept on what are called natural antibodies. And natural antibodies are actually really interesting. I studied them in graduate school and in my research, so I think they're really important. But this is a great example to teach about natural antibodies. So first of all, when you think about an antibody, you're probably thinking about a pathogenic antibody. So for example, if we're infected with influenza, part of our immune response would be to make antibodies to the influenza virus that help to eliminate it and prevent us from reinfection with that same influenza. So that's a pathogenic antibody because it's being raised in response to an infection. Natural antibodies are different. Natural antibodies are present, actually they're present in babies. They are present they're not associated with disease. In fact, if I were to test your blood in the studies that I did in my research lab, I would be able to show you that many of you have antibodies to different viruses that you've never been infected with. And so natural antibodies were kind of debunked. I mean, a lot of people thought they were insignificant. Oh, who cares about them? But we've now learned that they are really important in helping to shape our overall immune repertoire and helping to have a healthy, very well-rounded immune response. But why am I talking about them in context to the ABO blood typing? Well, this is how we learn how not to respond to our own red cells, to the carbohydrates that are on there. So when a person has blood type A, they have in their blood anti-B antibodies. These are not the natural antibodies. These are agglutinins. So that means that there's going to be a reaction. Now, the reason why people with blood type A don't respond to blood type or to one if they're given A blood is because they've tolerized. They've gone through that process of immune self-tolerance where they've learned not to respond to their own self, not to respond to self antigen. So we have natural antibodies to our own blood group antigens. So for me, I'm blood type A. So that means I have anti-A natural antibodies. Those don't cause me any problem at all. In fact, those are what are helping my immune system know not to respond to blood type A. But I also do have antibodies to B as well. But those ones didn't go through the process of tolerance like my A ones did, because I don't have B on my red blood cells. So there's no way for my immune system to become tolerized to B because I don't have that antigen in my body anywhere. But with A, it works. So we actually have antibodies, natural antibodies to A and B. All of us do. But those aren't the ones that are associated with transfusion reactions and with pathogenesis. Natural antibodies are completely healthy. The more well-rounded of a natural antibody repertoire that we have, the more healthy that person is. And we even get a little bit of cross reaction and protection from different viruses by having these kind of spontaneous antibodies in us. So a person that is blood type A is able to get blood type A or blood type O. They may not get blood type B because they have these agglutinins for B. They have anti-B antibodies in their blood, and they have the B cells that produce these antibodies, for the lifetime. So if they are given type B blood, these antibodies are going to clump onto it. Agglutinate means stick together. So notice that this is a pentamer. This is the IgM pentamer because it's got 10 binding sites on it. So that's a place to bind 10 different of these 10 different B carbohydrates. That's how agglutination happens. It clumps it together. Oppositely, a person that has type B, they have the agglutinous type anti-A antibody. So they're able to get type B and type O, but they can't have anything with A in it. Now, people with blood type AB, they can get anything. They are the universal recipient because they're tolerized to both A and B. They don't have any of these agglutinins in their bloodstream. And then type O, these are the universal donors. They can give blood that can be received by any blood group. See, type A can get O, type B can get O, type AB can get O, and of course, type O can get O. So that is how we prime and tolerize our own body to not respond to the carbohydrates that we have on our own red blood cells. So there is very much a difference between natural antibodies and between pathogenic antibodies. And if you're interested in learning anything more about natural antibodies, I do have a couple papers published on that topic. So anyway, maybe you'd be interested in looking those up. I'd be happy to send them to you if you're interested. Okay, so let's move forward. Now, in the United States, these are the frequencies of the different blood groups. So we're looking at a percentage, breaking it out by white, Black, Asian, and then Native American, and then we've got our 4 blood groups. So type O is high among the Native American population, and then it's pretty much the same amongst the other populations. Where look here at type B, it's the opposite. It's pretty low in Native Americans. And more frequent is found in Black and Asian Americans. So the reason why I bring this up is because sometimes it's a matter of having the right type of blood supply to match the demographic needs of the community. For example, when I lived in Arizona, we did a lot of medicine on the different reservations that we would drive to different Indian reservations and set up a clinic there for a day, and then we'd move to another one the next day, and we'd spend about a week out there. But we had to change the blood group, our blood supply that we had available with us for those trips because of the differences in the frequency of the different blood groups. So this is a picture of what agglutination and, well, non-agglutination looks like. So a person that is blood type A, so there is no reaction with A. So this would be A, this would be B, or it could be B. And then you can see, so this here is showing agglutination. So this means the person has pathogenic antibodies that are reacting with that blood. So I don't necessarily, in the PhysioX activity, I'll show you how to read them and how to interpret them. I've put this here more so you can see the difference between agglutination. So I'll point out all the ones that are agglutinated. We have it right here, 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 and here. So the ones where you see the more diffuse red are not agglutinated. Okay, so now let's get to the Rh factor, which is also known as factor D. So for example, I am A positive. That means I have A antigen and I have Rh antigen. So the ABO refers to your A or B positivity. In other words, if it says A, you're positive for A. If you have B in there, you're positive for B. The plus or minus at the end, that is the Rh factor or factor D, the rhesus factor. And it's positive in 80% of the US population. So it's no surprise that I have it because, 80% of the population has it. But people that do not have this antigen, the Rh negative people, they are at risk of developing an immune response if they are exposed to an Rh positive person. Well, that's pretty similar with the type A and type B blood as well. But where this does become a problem, and why it is dangerous is during pregnancy, if the mother and the baby have a different compatibility between their Rh. So if a person is Rh negative and they receive Rh positive blood, that is a life-threatening situation. So in pregnancy, the mother is tested for this, and there's actually medications that are given to mothers so that they can be protected from that potentially fatal agglutination that would happen that has the potential to affect both mother and baby fatally. And it was weird. A couple semesters, I had a student in my class who was Rh negative. And it was so weird. She knew her blood typing group, and we were doing this experiment over and over again, and it kept coming out Rh positive. And we later found out that she was pregnant at the time, and she did have exposure, and her baby was Rh positive, and she's Rh negative. So I don't know if I'm not saying that that we were able to determine that she was pregnant from looking at her blood typing group, but it was an interesting parallel that went along with it. She didn't know that she was pregnant, and when she found out that she was Rh positive, she went and got a pregnancy test because she had exposure during her previous pregnancy to her previous baby was Rh positive. So she got on the medication that ended up preventing this transfusion reaction from happening. So that's what the plus and the minus mean. So please don't sorry, I'm trying to, let me get a box up here. I can show you a little bit better. Here we go. So if we have a person that is A positive, B positive, we don't write it that way. So the way that we would write that is they are AB. That means A positive and B positive. Okay, so the positive and the negative simply refer to the Rh factor. That's all. Okay, and I lost my mouse, but I think we're on the last slide. Just a second, class. This happens it happens often, as you know. I stopped using the text box because of this. I don't even know where the prompter is. Oh, I'm getting there. There we go. Okay. Sorry about that. Okay, so here is the last slide. So sorry, it's a little blurry. So this is just another way of showing you who can give and who can receive based on blood typing. So a person that is O negative is able to, an O negative recipient can get blood from all of these options. So you can go through and read it. So now we've got the negative is the Rh negative, the positive is Rh positive for each of these conditions. So if we have a person that has, using my example, I am A positive. So that means I can get blood from somebody who is type A, but I can't get AB, right? Because I would react to the B part of AB blood. I wouldn't react to A because I know how to deal with that. Okay. So now we're going to, let me just switch over the share. I'm going to bring up the Physio X activities to do. It's actually just one activity. That way, for those of you who do not have PhysioX, you can see it done here. So it is in exercise 11, activity 4, the blood typing one. I'm actually going to reshare it because I want to make sure that it carries over to the opening of the activity. Sometimes it doesn't. Okay, so we've got our objectives. I'm going to just get to the questions. This is all written out in your lab exercise that was loaded up onto Canvas. Okay, so red blood cell membranes, let's see, which is the correct answer here, have antigens that specify the name of an individual. Well, that's just kind of silly. I don't have an antigen named Miranda on my red blood cells. Genetically determined agglutinins that can cause a life-threatening reaction. Agglutinins that specify that individual's blood type. There you go. That's the answer. The ABO and RH antigens can cause incompatible blood types. Okay. Present in the blood of a donor can react with antibodies in the blood of a transfusion recipient, resulting in clumping. Cause the most vigorous and potentially, this is all of these. Okay. Which of the following statements about blood types is correct? Let's see. A person with type A blood can have 2 alleles for the A antigen. So that would be IA, or 1 allele for the type A and 1 for type B. Well, no, because then they would be type AB. So that's wrong. A person with type B must have 2 alleles for the type B. No, because they could have that recessive allele and just 1 dominant B. A person with type AB blood has 2 alleles for A and 1 allele for B. Well, then they would be AAB. That doesn't even match genetics. A person with type O blood has 2 recessive alleles, neither A or B. That's the right answer. Okay, antibodies to A and B antigens are found preformed in the blood plasma. That's true. Not found preformed in the blood plasma. They are produced only after exposure to blood. They're present on the red cell membranes. Well, the antibody is not present on the red cell membranes. The antigen is, and they're not present in everyone. I don't have B antigen. Oh, wait a minute. This is antibodies to the A and B antigens are present in everyone. Yeah, that's true. But they're also preformed because they're part of the natural antibody repertoire. That's kind of a tricky question. I don't really like it. All right, let's go. Let's get to the experiment. Okay, so here's our setup. And this is similar to what you're doing with your blood typing kit. I'm not able to demonstrate that. That's kind of tricky to do with only me. I actually have to isolate myself from people right now due to some risks from COVID. I have a surgery coming up, so I just have to be isolated. So I can't have anybody come and help me do it. So I'll show it to you this way. But please feel free to do it at home following the instructions that are in your lab exercise. It's a pretty safe procedure to do. Okay, so we're going to begin by getting our tray that we do the reaction in, and we're going to put it on our workbench. There we go. And now we're going to get sample 1 and put a drop in A. Got it. Oh, and B and in RH. So we're testing sample 1. We've got 6 different blood samples that we're going to test. And now we're going to add the anti-serum, which goes in A. And then we have to put the B anti-serum in B. and the RH in RH. Great. And now we need to mix it up. So we're going to take the blue tipped one and put it in A. There we go, mix it up. It has to be a toothpick to do this. If you use glass, it actually can oxidize and mess up the results. So that's why the instructions do say to use a toothpick. So we're gonna do the yellow one goes in B. Get rid of it in biohazard. This is blood, so we do need to dispose of it properly in the biohazard container. There would actually be 2 layers of bags in that container so that if the inner bag pops, the outer bag is likely to be safe. Now we're going to take our tray and we're going to put it on a light box. So these just shine light up through the sample. Let's turn the light on and ta-da, here's our results. So we're going to go ahead and mark if it's positive or negative. So in the well with the anti-A, there is clumping. This is a positive result. But B is negative, negative result. RH is positive. And now we're going to record the data. Okay, and now get rid of this tray because we're done with it. Okay, if the patient's blood type is AB negative, what would be the appearance of the tray? So AB, so A would be clumped and B would be clumped. And then the negative means the RH is negative. So that part would be unclumped. So it would be A clumped, B clumped, RH unclumped. Okay, so now we're going to finish up the rest of the samples. And fortunately, PhysioX does this for us quite a bit. See, also, I don't know if you've noticed, but PhysioX had an upgrade. I'm so excited. It doesn't make me have to hit the next button on every single step now. It knows when it's done it and tells you and advances it. So yay, simple things in life. Okay, let's record our data. So A is negative, or anti-A is negative for A. This is positive and this is positive. We're going to record our data. Move on to sample 3. Whoops. Of course, it would help if I got the plate. There we go. So the materials that we use in the lab are actually chemicals that do this. We use our own blood, which is cool. But the anti-A serum is something that we purchase from a supplier. We used to only do this experiment using dog blood. I don't know why. I guess they just didn't want to use human blood. But all of our labs are able to use human blood, especially when it's your own blood. So that's pretty neat that you get to, I think that you have the option to do this on your own. Okay, sample 4. If we were meeting in lab, we would also be doing the BioPak measurements on your own self. So you'd have quite a bit of data at the end of the semester. Okay, these are all negative. Let's get sample 5. Positive, positive, positive. And now our last sample, blood sample 6. And then we'll go through and record the blood type. So one thing that students often ask me with this experiment, but also with others, is, well, how do I know if my sample has a positive result? And my answer is pretty much the same every time. And it is there a change from the negative control result? So I would be doing this with known samples of blood so that we could confirm that the experiment's working right. So when we're meeting in the classroom, students do their own blood, but I run known samples of blood. So I have a sample of blood that's known to be A, known to be B, and so on. So that serves as the control for it. All right, now we're going to go through, sorry, the alignment on PhysioX is pretty bad. It doesn't let me resize the view. So we're going to go through one by one. So that means positive with A, anti-sera, negative with B, positive with RH is A positive. A positive. That's how you would record their blood group. So we're negative on A, we're positive on B, we're positive on RH. So this person is B positive. Now we've got positive, positive, negative. So they're AB negative. Oops, let me scroll down. Okay. And now we've got negative, negative, negative. Ooh, so they are O neg. Their blood's in high demand. Positive, positive, positive. That's AB positive. And then sample 6 is negative, positive, negative. That is B neg. Okay. So that is the completion of the actual experiment, but let's go through the questions. So why are individuals with the AB positive blood type known as universal recipients? Well, this means that they've had exposure to the A antigen, to the B antigen, and to the RH antigen. Their immune system has had the opportunity to self-tolerize against the antigens that are expressed on the surface of their own red blood cells. So therefore, they don't develop pathogenic antibodies that agglutinate. Instead, their immune system gets the signal that says, hey, that antibody to A that I have on my red cell, not a problem. We don't need to worry about you. The antibody is still there. It is produced, but it does not agglutinate. Natural antibodies have the same structure as a regular antibody, but they are made of different pieces, is a simple way to explain it. Okay, so they have neither A or B. Well, that's wrong because they have both. Although their blood serum contains, that's wrong. They have both A and B antigens, that's true. And their blood serum does not contain antibodies against, wait a minute. Okay, so they have, the way I would write this on an exam, just so we're crystal clear, they have both A and B antigens on their surface of their red blood cells, and their blood serum does not contain agglutinous or pathogenic antibodies. So this is the right answer here. Is there any more? I thought there were some more questions. Okay, good. Okay, so now, now it took me back to the beginning. Well, there were a couple more questions, but I'm not going to go through the whole thing. It really just was a way to verify that the answers that that were submitted already were correct. All right, well, I hope that was helpful. Have fun with your blood typing experiment, and please reach out with any questions, and take care of yourselves. Bye-bye.